Welcome to Learning Science is Fun. I'm Jemina Camille and today we will learn about ceruloplasmin. Ceruloplasmin is a serum peroxidase that is responsible for 90% of copper transport. It stores and carries the mineral copper around the body, and it is well known for its role in the pathogenesis of Wilson disease. Copper plays an important role in your body's development, health, and functions, making ceruloplasmin, the protein responsible for copper production, indeed vital. Ceruloplasmin production occurs in the liver, and this peroxidase enzyme is encoded by the CP gene in humans. Liver hepatocytes mostly synthesize ceruloplasmin. Ceruloplasmin is not a routine value measured in standard labs. It is an acute phase reactant and may become elevated when someone has any form of trauma, tissue damage, infection, or inflammation. It bears mention that ceruloplasmin may be high in states of high estrogen and progesterone, which means pregnant women or women on oral contraceptives may have high levels of ceruloplasmin. Many medications, most notably the anti-seizure drugs like valproic acid, are also known to increase levels of ceruloplasmin. As stated, ceruloplasmin is not routinely measured, but the following symptoms may prompt ordering of ceruloplasmin levels. When it comes to the structure of ceruloplasmin, it has a molecular weight of 132,000 Dalton and has a polypeptide chain of 1,046 amino acids and 3 asparagine-linked oligosaccharides with a total carbohydrate content of 10%. The crystallographic 3D structure of protein shows its 6 structural domains with a 3-copper catalytic center that is crucial for its oxidative function. Ceruloplasmin, a copper-containing alpha-2 globulin synthesized in liver and secreted in plasma, plays a role in several biological functions including copper transport, coagulation, angiogenesis, peroxidase activity, and defense against oxidative stress. This protein has both pro-oxidant and antioxidant properties and has been described as a moonlighting protein due to its many and varied activities. Ceruloplasmin antioxidant function is mainly related to its peroxidase 1 activity as well as glutathione peroxidase activity. By oxidizing ferrous iron to the ferric form, ceruloplasmin promotes iron loading onto transferrin, which binds only the ferric form of the metal. Oxidizing ferrous to ferric iron also serves an antioxidant function and thus helps prevent or reduce oxidative damage to proteins, lipids, and DNA. Ceruloplasmin measurement is via a blood test, which often occurs by analyzing a serum sample with immunoassays, immunoturbidimetry, or immunoephilometry. The sample needs to be spun and separated as soon as the laboratory receives it and assayed promptly. The serum test is often part of the workup when there is suspicion of Wilson disease and is commonly ordered along with urine copper levels. Lower than normal ceruloplasmin level may indicate Wilson disease, Menkes disease, copper deficiency, aceruloplasminemia, or in general states of low protein intake such as malnutrition, whereas greater than normal ceruloplasmin levels may indicate or present in copper toxicity, zinc deficiency, pregnancy, oral contraceptive use, lymphoma, acute and chronic inflammation, rheumatoid arthritis, angina, Alzheimer's disease, schizophrenia, and obsessive compulsive disorder. Among the genetic disorders associated with copper deficiency caused by mutation in the CP gene are the Wilson's disease and aceruloplasminemia. In Wilson's disease, copper is not put in ceruloplasmin. The disease also keeps your liver from sending extra copper to be eliminated in bowel movements. Instead, copper builds up in the liver until it overflows into the bloodstream. From there, copper builds up in your brain, corneas, kidneys, liver, bones, and small glands near the thyroid. If not treated, the liver and brain damage due to copper poisoning from Wilson disease is fatal. One clear sign of the Wilson disease is having Kayser-Fleischer rings, which are brown rings around the corneas in your eyes. These rings are seen only through an eye exam and happen in 98% of people with Wilson disease who have nerve symptoms, and they happen in about 50% of people with liver symptoms. On the other hand, aceruloplasminemia is a rare adult-onset autosomal recessive disorder characterized by systemic iron overload due to mutations in the ceruloplasmin gene, which in turn lead to absence or strong reduction of ceruloplasmin activity. Ceruloplasmin is a peroxidase that plays a key role in iron export from various cells, especially in the brain, where it maintains the appropriate iron homeostasis with neuroprotective effects. The main clinical features of fully expressed ceruloplasminemia include diabetes, retinopathy, liver disease, and progressive neurological symptoms reflecting iron deposition in target organs. That concludes everything you need to learn about ceruloplasmin. To learn more about other proteins and their functions, check out our website at bsedsciencepup.com because learning science is fun.